Hello, everyone, and welcome to today's webinar, Identifying Blood-Based Signatures of Traumatic Brain Injury Using High-Sensitivity Multiplexed Protein Assays. My name is Delia Osborne with Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Alamar Biosciences. To learn more, visit alamarbio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd now like to welcome our speakers for today's webinar. Alex Forrest-Hay, Senior Vice President of Sales, Alamar Biosciences. Dr. Cheryl Wellington, Professor and Vice Chair Research. Javad Mofagian, Center for Brain Health, Pathology and Laboratory Medicine, the University of British Columbia. And Professor David Sharp, Associate Director of the UK Dementia Research Institute and the lead for the UK DRI Care Research and Technology Centre, Imperial College London. You may now begin your presentation.
Hello, everybody. Thanks for the introduction. My name is Alex Forrest-Hay. I'm the Senior Vice President of Sales at Alamar Biosciences. I'm thrilled to be here today with Professor Wellington and Professor Sharp to talk to you about our new ELISA technology and its application in traumatic brain injury. So the title of my introduction will just be New ELISA, fully automated, ultrasensitive proteomic biomarker platform for profiling biofluid samples. So Alamar was founded in 2018, and our mission is to power precision proteomics to enable the earliest detection of disease. And we've pioneered a new technology in the proteomic space that provides unprecedented sensitivity, meaning you can detect proteins and protein biomarkers from blood and other fluid samples at the attomolar level, which is down in the femtogram or sub-femtogram per mil levels. So this means you can now quantify very low abundant proteins and detect biomarkers earlier in the disease progression. But as well as high sensitivity, New ELISA also offers unprecedented dynamic range. So you can look at proteins across 12 logs of dynamic range, which is absolutely critical for plasma proteomics in particular, where proteins can be in the blood at very varying concentrations. When you think about traumatic brain injury, for example, a protein like GFAP could be extremely high after an injury, and being able to measure across this very broad dynamic range enables you to look at very large numbers of proteins together simultaneously. The new ELISA platform also has very high levels of flexibility, so we can build broad panels of biomarkers to look at things like inflammation, or broadly look at neurodegenerative biomarkers, or you can develop singleplex or small groups of markers if you want to develop diagnostic panels or single assays. And what we've also done with New ELISA is we've fully automated the platform, so you can run it on our Argo instrument with very few hands-on steps and very high reproducibility. This was all published in a paper in Nature Communications in 2023, and you can see the link here. So I'm going to talk a lot about sensitivity, but it really is critical. And when you think about lowering the limit of detection so that you can now detect these critical biomarkers much earlier in disease. And you can also measure healthy baseline of samples that maybe are pre-symptomatic or ostensibly healthy from a symptomatic standpoint, but actually are starting to get the early signs of disease. So what New ELISA does is improve sensitivity of detection in blood by thousands of fold over traditional technologies like ELISA assays. The Argo instrument, or the Argo HT, is truly proteomics at the push of a button for the first time. So everything is provided in a consumable kit, which is lyophilized. So all the reagents that you require to do a run of 86 samples are provided in a simple kit and shipped at room temperature. The Argo HT system is a benchtop instrument, which is capable of running 3 96-well plates in a 24-hour period. There's an onboard qPCR instrument to do the readout, and also the instrument will do all of the steps of the assay automatically in about 8 hours per plate. We also provide software, which is either cloud-based or on-prem software, to allow you to do both the running of the instrument and also downstream analysis, statistical analysis, volcano plots, etc. So you can very quickly go from sample to data in less than 24 hours with very few hands-on steps. The concept here is to be able to go from discovery of signatures of biomarkers right through the translational process to create your own diagnostic assays with the same high sensitivity assay and a seamless workflow on the same instrument. So we use monoclonal antibodies, which enable you to actually look at hundreds of biomarkers simultaneously in a panel, and then translate those into smaller panels, and then use the qPCR readout for looking at individual biomarkers or small subsets of biomarkers for diagnostic applications of the future. So how does it work, and how do we achieve such high sensitivity? Well, the NuLISA technology is really based on a simple premise that if we can remove background, we can significantly improve signal to noise. So we've taken a technology known as proximity ligation assay and evolved it and added some additional purification steps that allow us to achieve unprecedented levels of sensitivity. So here in this schematic, you can see that we use two antibodies, monoclonal antibodies, as I mentioned, that bind to two different epitopes of the protein that you're trying to measure. So these two binding events have to occur simultaneously to actually create the immune complex and ultimately the signal. So to these monoclonal antibodies, we conjugate DNA sequences or oligos sequences with a known barcode. So we can essentially use either qPCR or sequencing to do the readout by attaching DNA molecules to the antibodies. What you can see on this schematic is that we also have double-stranded sequences. On the first antibody, we have a double-stranded sequence that ends in a poly-A-tail, and on the second antibody, we have a double-stranded sequence that ends, which is biotinylated. And what this enables you to do is capture the immune complex onto magnetic beads and do sequential wash steps, which remove background and improve sensitivity significantly. So the steps of the assay are outlined here, and all of these steps occur automatically on the Argo instrument. So the first step is really just to integrate the conjugated antibodies into your plasma or CSF sample, and they will bind to the protein of interest and form this immune complex where the oligos stop in close proximity to one another. From there, we add oligo-DT beads into the sample so that the poly-A tail on the first antibody binds to the beads and captures the immune complex. And this allows the instrument to then do a simple wash step to remove any unbound antibodies or proteins that aren't part of the panel or assay that you're trying to measure. We then release this immunocomplex back into solution and use streptavidin beads, introduce streptavidin beads to use the strong bond between biotin and streptavidin to capture the immune complex in the inverse direction. So we now have a second capture step, which we then can do a second wash step to remove background further and essentially purify that immune complex. So once this is done, we can then perform the molecular biology by ligating the ends of the oligo sequences together, and then measure the protein either by using qPCR for the readout for small plex panels and single plex assays, or by integrating a sample barcode and utilizing next generation sequencing from a pooled library, so we can look at hundreds of proteins simultaneously in 86 samples from a plate. So by doing this, we leverage the latest DNA technology along with this proprietary background suppression to drastically improve sensitivity and improve the precision of the proteomics assay. And so this was showcased in our Nature Communications paper. On the left-hand side, you can see an IL-4 assay, and the blue line represents the NuLISA technology, which gives a limit of detection down in the attomolar level, and also a dynamic range across about 8 logs. And so when you compare that to the red line, which is a traditional ELISA or proximity ligation assay without the background suppression, you can see a 10,000-fold improvement in the limit of detection. And this is using the same antibody pair. And on the right, you can see a comparison with the previously best-in-class technology from Quanterix called Señora for a HIV P24 assay, which shows that Neulisa is 10 times more sensitive and has a broader dynamic range, but whilst using less sample input. So we've developed many hundreds of assays now using Neulisa, and one of the most exciting aspects of the technology is that you can do single readouts with high sensitivity or multiplex readouts using the NGS with the same sensitivity. And again, this data from our paper showcases 5 cytokines that are measured either with the qPCR singleplex or the NGS multiplex readout, and essentially show the same sensitivity and a really high correlation between the 2 readouts. So this gives us the ability now to build very large panels, such as this CNS disease panel 120, which allows us to capture all of the key markers linked to brain health and neurodegenerative disease in a single assay, which can utilize as little as 10 microliters of plasma or CSF. So what you can see here is that we can measure beta amyloid, phosphorylated tau. You can also measure other markers linked to other things like Parkinson's disease, like alpha-synuclein, or ALS, like TDP-43, as well as critical biomarkers that have been seen in traumatic brain injury, like NFL, GFAP, and UCHL1. And so this panel is really changing the game in terms of understanding all the hallmarks of CNS disease or brain health, which can be applied to really a whole range of different neurological diseases. And this panel allows you to look at 120 biomarkers from 10 microliters of blood, and will be showcased by David and Cheryl in their publications. The panel has also been used extensively in Alzheimer's research by some of the world's leading labs, including the University of Gothenburg, Banner Health, University of Pittsburgh, and Washington University. And these publications that you can see referenced here all showcase the ability of the CNS panel to perform as well as singleplex clinical assays for understanding things like p-tau217 in Alzheimer's disease patients and how that correlates with amyloid PET, for example. So well worth reading these papers to understand the power of using this approach to really have a more holistic view of Alzheimer's disease and the underlying pathology. And what we're really excited about is the ability to now differentiate between patients who may come to a memory clinic with mild cognitive impairment and those that have actually got much more advanced disease, like those with early tau deposition. And what you can see here is a volcano plot that shows that these 2 patients, in this case, have very different profiles when it comes to the proteins in the blood. And we can now measure all of these with the Neulisa assay in a single experiment and get much more clear, comprehensive signatures of different stages of dementia or neurological disease. We're also very excited about a recent development where we've developed an assay for brain-derived p-tau217, which actually appears to outperform traditional p-tau217 assays when it comes to diagnosing Alzheimer's disease, mainly because what you're actually measuring with this brain-derived tau is the tau that's building up in the brain rather than the potential for total tau that may come from the periphery. We believe this is going to be very important again in differentiating different neurological diseases. And we've demonstrated this in an ALS cohort where we've seen that p-tau217 from the brain is actually not as upregulated as the p-tau217 from the periphery in the ALS patients. So again, giving a much more defined insights into the patient and the opportunity for better diagnosis. So just to wrap up here, we also have a very powerful inflammation panel, which contains 250 biomarkers really linked to immune health and inflammation. The most comprehensive view of cytokines and chemokines on the market. We can measure all 250 of these biomarkers from 10 microliters of blood or CSF across a very broad dynamic range. And as you know, inflammation is a key driver of neurodegenerative disease. And I think this will also be showcased in some of David's work after my presentation. But what's amazing about this panel is we can truly measure all of these 250 biomarkers in the plasma of healthy individuals as well as disease individuals, which has been a long-running challenge as cytokines tend to be very low abundant in blood samples. One final thing to mention is that we've actually tested and validated this platform now also on microsampling devices. So we're really excited to be able to, potentially go into, the field and pull samples out in real time, put them onto dried blood spots, and then utilize our panels to actually measure all of these biomarkers from dried blood spot samples and remote blood collection. I think this is going to enable us to do real-time diagnosis much faster in the future, and also enable us to get into populations that are not able to go to a phlebotomist or a clinic. So this is a really exciting development and another major advantage of the high sensitivity that NULISA brings. So just to wrap up here, we have an ever-expanding array of different assays. We have the inflammation panel that I mentioned and the CNS panel for brain health. We've also developed a mouse panel, which means you can do reverse translation of the findings from human into mouse, utilizing very broad content for inflammation and CNS diseases. And we have a range of singleplex assays, which is growing as we develop more singleplex assays using the qPCR readouts. And just to mention, the mouse panel is a combination of immunology markers and neurology markers and oncology markers that can be really helpful in terms of understanding the brain health pathology or the immune profile in small sample sets from mice, whether it be CSF or plasma. And we recently launched the ability to develop your own NULISA assays with our custom assay development kits. So now if you have an interesting biomarker of your own, you can utilize the high sensitivity that NULISA brings in your own lab by developing your own assay using the NULISA chemistry. So just to wrap up, the product menu we currently have is the multiplex panels that I mentioned, the singleplex assays, the ability to develop your own homebrew kits, and then we also offer services through our lab in Fremont, so you can leverage the technology and generate the data yourself, which is indeed what Professor Sharp did for one of his studies. So I'll wrap up there, and I'll hand over now to Professor David Sharp from Imperial College in London, who's going to showcase some of the work he has done utilizing the NULISA platform. Thank you.
Hello, everybody. My name's Dave Sharp. I'm a neurologist based at Imperial College London, and I'm ringing in from London. I'm going to give you a brief outline of our work looking at NULISA analysis of traumatic brain injury bloods, and explain to you why this is a very interesting direction of travel going forward. So the work relates to a particular paper that's already published in Brain. You can see here. So if you're interested in more detail, it's all or the majority in this paper. So we're going to focus on traumatic brain injury. These are very common. These are resulting from road traffic accidents, falls, sporting injuries. And they're a very large cause of morbidity and mortality within the world. And we have a kind of challenge of understanding them, so partly diagnosing them, but particularly making prognosis about outcomes after traumatic brain injury, because they're extremely complex in nature. I'll go through that. So if we think about the kind of long-term effects of traumatic brain injury, they're widespread. Things like cognitive problems, behavioral difficulties, psychiatric issues, epilepsy, movement disorders, migraine, functional problems, vestibular disorders. And the challenge is that the mechanisms by which the brain is injured are very diverse. So, for example, we might see brain contusions or diffuse axonal injury, the triggering of neurodegenerative pathologies, the triggering of neuroinflammation or metabolic problems, and then associated cognitive difficulties and vestibular problems. And we have a great challenge in terms of identifying these mechanisms and pathologies, which means that it's often a struggle to make a good prognosis or to work out which treatments need to be given. So our current kind of approaches to diagnosis are largely imaging based. So we're using CT imaging, but also MRI and sometimes PET imaging to look at the different injuries happening in the brain. But there's an enormous opportunity with fluid biomarkers to identify the presence of different kinds of pathology, and that's what we're going to be focused on. And that's where I think the NULISA technology could be transformative. So I think the challenge is that traumatic brain injury is complex and a mixture of different kinds of brain injury. We often miss subtle injuries, and clinical outcomes can be difficult to predict. And our treatments are limited, particularly pharmacological treatments have not really developed successfully. And that's to a large extent, I think, due to our inability to identify specific treatments with accuracy. So the opportunity, I think, is that the whole TBI field is moving towards a more biology-based classification. So what I mean by that is that we want to be able to classify, as in neurodegenerative diseases, the underlying pathologies like the presence of axonal injury or the presence of inflammation. And to do that, we need new tools that can be used at scale. And I think the new ELISA technology is very well placed to transform a number of areas, for example, screening and monitoring of patients. So if we think about the use of fluid biomarkers, this has been a developing field for some time. This is a figure from Henrik Zetterberg's paper, one of his papers, and Henrik and I collaborate on much of this work. And what you can see in this figure is the kind of range of different signals or different proteins that are released from a damaged cell and associated glia, which can be picked up in the blood. And the focus to date has been around things like neurofilament light, GFAP, tau species, amyloid and cytokines, and all of those have been shown to be affected by traumatic brain injury, and to a greater or lesser extent kind of useful in terms of understanding the effects of injury. And the big step forward in recent years has been the ability to use highly sensitive blood-based biomarkers, and that's where I think ELISA comes in. So ELISA gives us the opportunity to measure not just the traumatic brain injury direct kind of markers like NFL and GFAP, but also the associated neurodegenerative markers and also the inflammatory markers, all within the same panel. So I think in terms of kind of the targets moving forward, I think there are big opportunities in a number of areas. One of which is in screening and the diagnosis of injury. So particularly on the mild TBI side, there may not be obvious imaging changes, and most frequently there aren't. So one thinks about sporting injuries. They may be very symptomatic, but usually the imaging is negative. Therefore, in terms of screening and the identification of the presence of a brain injury, there's a great utility for a sensitive blood-based biomarker that would pick that up. In terms of prognostication, we're not very good at prognosis after TBI. So there's a great opportunity there to improve prognosis through the use of blood tests. We want to be able to kind of measure the progression of the disease itself. It's clear that TBI is not just a static insult, but it produces a kind of reverberation over the months and years in a range of different pathological processes that we can measure using these blood tests. We want to be able to measure the effect of different treatments, so measure new therapies. And then importantly, in parallel, the ability to do discovery science work with an aligned panel that's linked closely with the patient clinical work is hopefully, I think, going to be quite transformative. So what we can see here is some data looking at the effects of mild traumatic brain injury on plasma biomarkers. And what you're seeing in yellow is UCHL1 and in blue GFAP. And if we look at the bottom panel here, this is related to mild TBI. We can see a big rise in GFAP in blue and an initial big rise in UCHL1, which gradually reduces over a number of hours. So we're getting a big signal in the blood from even mild traumatic brain injury. We've looked at this in a study that I led called BioAxe, which is a big European study that involved a number of European centres. And the data I'm going to present to you today is from this study. So this is a little bit of work from using Quanterix Señora that I'll use as a comparator. We can see very high levels of NFL, total tau, UCHL1 and GFAP. In red, it's in the blood, so this is plasma measurements. But in blue, we have microdialysis measurements. So these are samples taken directly from the brain of patients who've been injured, who've had quite severe injuries. And the important thing to note here is that there's quite a nice correlation between the levels in the blood and the brain. So the blood is giving us pretty good readouts of what's happening in the brain. So on to the ELISA work. So I'm going to present some data on 86 patients who had moderate to, or 38 of them had moderate severe TBI. We have a group of non-TBI trauma controls. And that's important because we're interested in what's specific about traumatic brain injury, not just the general effects of trauma, and then some non-injured controls. These are blood samples that were taken in the first 1 to 2 days after injury, so quite acute. And then I'm going to present mainly the work on the CNS panel. We've also done some inflammatory panel work as well that I'll just touch on at the end, but the majority of the work is just around the CNS panel. We also have imaging and a range of other measures, and I'll touch on the relationships between the new LISA results and the imaging, which are also interesting. So one question we were interested in was what was the pattern of proteomic changes after TBI that was specific to brain injury. And what we're showing here in a slightly complicated slide on the left are volcano plots that relate to what's higher going from left to right. And then the probability, the p-value, is on the y-axis. So the way to read this is, for example, the top panel is showing the contrast between traumatic brain injury and controls. And what we can see up here in the top right-hand corner, although a bit small, is that GFAP is both very highly increased, a very large amount of increase, and it's a very significant difference between TBI and controls. So this allows us to compare and contrast between our 3 groups, between the TBI group, the control group, and the non-trauma controls. And we can turn that into this Venn diagram, which is, I think, quite informative. And the way to read this is the TBI specific markers. So anything that's elevated in a TBI specific way is in this quadrant down here, marked TBI specific markers. And we can see a range of different proteins. For example, GFAP, which is what I've mentioned before, is elevated there, S100B. But we also have a range of other markers that have not been described. And one of the things that the new LISA panel allows us to do is to look in a more kind of discovery context, a whole range of markers that have not been previously explored. And I'll talk a little bit more about that as we go on. In the middle, we have more general injury markers that are exacerbated by traumatic brain injury. And in the top panel here, we've got proteins that are non-specifically increased after trauma. So we can see things like CRP are increased in this group, which is not surprising, but it's not something that's specific to TBI. Okay, so this is replotting that data. So what we're doing here is plotting the proteins that show differential expression such that they're higher specifically in TBI. So we have neuronal markers, neurodegenerative markers, astroglial markers, markers of tau biology, markers of cytokines and chemokine response, and then a marker of cell energy metabolism. And all of these are, really interesting in the context of traumatic brain injury. And so if we look at 1 or 2 of these, if we look at GFAP here, GFAP is highly increased in the traumatic brain injury group, which is a kind of sanity check, really. We're seeing something we really expect to see, but very sensitively expressed in the panel. So that's really really good. If we look at UCHL1, which is the other thing that I showed you about already, again, we see high levels of UCHL1. We weren't measuring in the first hour or 2, but which is probably why it's not higher. But still, we're very sensitive at picking that up. But we also see a number of other, really interesting things. One of the things we can look at with new LISA is various aspects of tau biology, which are really interesting in the context of traumatic brain injury. So, for example, one of the phospho-tau species, 231, shows differential expression, so increased levels in traumatic brain injury. MAPT levels, a kind of marker of total tau response, is also elevated. And I know in the new version of the panel, we've got a range of different brain-derived tau species, which I think will be really informative at disentangling what's peripheral and what's central in the response to injury. And then we have a kind of range of cytokines and chemokines that are also expressed in interesting ways after TBI. Here we can see IL-16 elevated and CCL2, both cytokine responses, perhaps partly mediated through microglial activation. So we can look at the relationships between the different biomarkers. Here we're showing correlation plots between the range of biomarkers that are increased after traumatic brain injury, and this gives us ideas of how these things are interrelated. If we look at GFAP, for example, we see quite strong relationships between GFAP and the tau biology kind of markers, as well as GFAP being associated with IL-16. So the ability to look at this in a single platform allows us to unpick the interrelationships between these different types of markers from different pathways, which I think is particularly informative after traumatic brain injury. We can take the whole panel and cluster in a data-driven way. So on the y-axis down here, although you won't be able to read it, we've got all the different markers from the 120 CNS panel. And in a data-driven way, we can cluster these depending on the expression of the different markers. And by doing this, we produce three clusters 3 to 5, that are very TBI heavy. So the red is TBI, and you can see most of the TBI falls within these clusters. And having done that, you could ask questions about how those clusters relate to biology. So on the right, what we're seeing is that the TBI specific clusters are related to lesion volume. Now, this isn't surprising. So the amount of lesion within the brain from the MRI imaging here is very closely related to different clusters, but it does show how we can begin to kind of relate this detailed mapping of proteomic response to biology that's picked up by imaging and by other measures. So a key thing that we're interested in looking at in TBI is the damage to the white matter. So the head injury itself and the biomechanical forces will damage the white matter. What we see here is a snapshot from a diffusion tensor imaging scan, which has been a transformative technology to allow us to map out the location of white matter injury. And when we look at the location of white matter, this is a map of different areas of white matter damage, where white matter damage is seen in red. We can see that there's plenty of white matter damage in our group after the injury, but the interesting thing is we can relate specific proteomic signatures to the pattern of white matter injury. So, for example, we can see that UCHL1 is correlated with the amount of white matter injury. More UCHL1, more white matter injury, which is interesting, not entirely unexpected. But then we see some other very interesting markers. So, for example, CQS T, so sequestome, is a marker of autophagy, which has never really been investigated in traumatic brain injury. And here we're seeing quite a nice relationship between the amount of autophagy triggered TBI and the amount of damage to white matter within the skeleton. So I think we're beginning to kind of identify completely new relationships between the pathophysiology of the injury identified in the blood and other biological signatures, for example, in the imaging. A little bit more on cytokine response to white matter injury. Again, similar kind of idea. We're mapping the cytokine response to the presence of white matter injury. And here we're seeing relationships between a number of cytokines. For example, IL-6, which has been a particular target for treatment after TBI, and IL-33. And again, if we're looking at longer term outcomes, the GOSI is a functional marker of outcome at 6 months and 12 months. So higher level of GOSI is associated with better outcomes. And here we're seeing relationships between IL-33, one of the IL-1 family cytokines, and functional outcomes. So again, this is suggesting that these cytokine levels are going to be useful in terms of predicting outcomes. We've also looked at the relationship between the CNS panel and the inflammatory panel, and either ELISA or Olink measures. So we're interested in to what degree there's a tight correlation. In general, there's a good correlation between the different types of assessments. So we can see, for example, for IL-6 and for NFL, there's very, very good correlations. That isn't true across the board. So IL-4, for example, measured by the CNS panel, is not at all well correlated. And similarly, IL-7, IL-18 doesn't show a particularly good correlation. So there's some complexity about the reason for this variability and how much one can map from one type of assessment to another. So just to finish off, I do think there's a major opportunity with the use of the new ELISA technology to look across a whole range of traumatic brain injury. And the attraction here partly is that we have a lot of very well phenotype studies with banked blood samples already. So this work can actually be done very quickly. And we can look across different types of TBI. So, for example, civilian, military, sporting TBI. We can look at different severities. I've talked to you a bit about the differences between mild and moderate severe injury, but different mechanisms. So if there's focal injury, diffuse axonal injury, blast exposure, these are all likely to have a different proteomic signature that we need to define, and also different timings. I've talked to you mainly about the very acute assessment, but the way that secondary injury evolves over time in the subacute phase is extremely important. And then I'm particularly interested in the long-term chronic effects. So the link between TBI or repeated TBI and dementia is a very important area that we can begin to unpick using the new ELISA technology. So importantly, we need to link this work with preclinical and discovery science models. So the availability of a matched or linked rodent panel is potentially extremely valuable. And if we can get to the point where we can do aligned human and animal studies and begin to understand the kind of mechanistic relevance, particularly of new treatments, I think that's going to open up new doors for TBI research. And then we could do complementary analysis from a range of other disease areas, which again, a kind of transdiagnostic approach here, comparing the signature of TBI to, for example, neurodegenerative markers in AD or other pathologies, other dementia pathologies, will be very informative. And just to return to a slide from the start, I think the value of all of this from a clinical perspective, I'm a neurologist, so I want new tools to use. I want to be able to use these tools, ideally perhaps a cut down panel for screening of TBI, for diagnosis, for prognosis, for measuring disease progression, for measuring the effects of therapies, and also aligning that with discovery science work with kind of linked rodent panels. So I will stop there.
Hello, everyone. Show of gratitude to Alamar for being able to host this meeting and offer me the ability to present today. So I'm going to be talking about our experiences in using blood-based biomarkers for brain injury. And it's going to be a fairly broad coverage of all of the activities that we're doing in this domain. These are my disclosures, mostly grant funding, that some of which is relevant to the work that I will share today. So currently, neurological conditions are assessed in the clinic using questionnaires, clinical exams, or neuroimaging. With respect to laboratory medicine, cerebrospinal fluid has historically been the matrix of choice for obtaining fluid that directly contacts the brain and spinal cord, from which there are several approved tests for a number of different neurological conditions. But of course, using blood samples has enormous potential to broaden the use of biomarkers and enabling greater representation and participation at a much more cost-effective methodology. There are several fluid biomarkers, mainly proteins, that have undergone an extensive array of research studies for a variety of neurological conditions that most of us here are familiar with. And I will be speaking about many of these biomarkers in the presentation in the context of their changes within brain injury. So when we think of brain injury, we typically first think about traumatic brain injury. So this is defined as a mechanical cause of deformation of brain tissue, whether it is a physical impact or a pressure wave. With Dr. Jamie Hutchinson, we have been working on the CAN-TBI program, which is Canada's flagship study of traumatic brain injury, very similar to the TRACK-TBI study or CENTER-TBI studies in the United States and in Europe, respectively. So several years ago, we published with Jamie an analysis of pediatric traumatic brain injury biomarkers. This was published using the Quanterix platform, where we first determined the reference range for total tau in pediatric specimens using samples obtained from what is known as the caliper cohort. So when we measure total tau levels with comparing to control patients, we can see that we have discrimination between severe and mild TBI. And when we split the mild TBI into those that had a perfectly normal Glasgow Coma score of 15 from those that had 13 or 14 scores, we can also see that total tau is slightly elevated in those with an altered GCS score. Furthermore, total tau was able to discriminate those patients that were CT positive compared to those that had a normal CT scan. Furthermore, the final thing that we observed in the study is that for children with TBI, those that were mild or moderate, serum total tau levels returned to normal within a few days after their injury, whereas in the severely injured children that remained hospitalized, they were both highly elevated and reduced compared to the control levels. This study was completed in 2019. Now, clearly the biomarker field has moved on since then, but we still have the major asset of the CAN-TBI biobank that includes not only children, but adult samples with TBI of severity of all severities. So we are looking forward to deploying Alamar into this biobank and look forward to sharing those results as soon as possible. Now, moving into preclinical animal model studies, in 2014, my laboratory, in collaboration with Dr. Peter Krypton, a mechanical engineer, developed and launched what is now known as the Chimera platform of murine traumatic brain injury. This is a closed head injury impact acceleration model that is now recognized to be very valid in terms of its representativeness to clinical mild injuries, especially such as concussion. So in the system, an animal is impacted just on a completely closed head. The head is absolutely free to move in space. And we've done many studies using the Chimera platform that have described its neuropathology and behavioral deficits. Here, what we're showing is some results using both Quanterix and mesoscale discovery assays, showing the potential for blood-based biomarkers to be performed in murine studies, where we can see that if we increase injury severity, we have a change of loss of writing reflex, which is the equivalent of loss of consciousness in a clinical study. We also see an injury-dependent, dose-dependent increase in plasma glial fibrillary acidic protein, as well as neurofilament light. Now, it's been, up until now, very challenging to routinely incorporate blood-based biomarkers into animal model studies, mainly for two reasons. First, antibody cross-reactivity may limit the number of tests that were designed for use in humans to translate into mice, just because the antibodies may not cross-react with the target of interest. Second, the volume limitations and cost considerations are substantive in murine studies. So here's where we're really excited to be able to now investigate the potential of Alamar for doing translational research in animal model studies. So we're still fairly new to the Alamar platform, and what I'll show is some very early work in progress slides that show some of the things that we are thinking about and our progress in what can the Alamar platform do in mice. Here, what I'm showing is our analysis of the detectability of targets for murine plasma, and you can see very good percent connectivity detectability. There are only a few targets that are really undetectable in murine plasma, so it's working as advertised, and we're very happy about that. When we do a chimera TBI study in mice, here this is using a tau model known as transgenic 4510, one of the p-tau models. Here we can see changes this is 6 months after injury. Now, this is still very early studies with only an N of 2 mice per condition, so we still have a long way to go. But if we bump up the false discovery rate to 50%, we actually see several biomarkers that remain lower after 6 months after injury, suggesting that there's ongoing changes after a traumatic brain injury in animal models that might be sensitive to blood-based biomarker detection methods. We are, of course, continuing our pilot studies in acute brain injury in mice. These were just convenient samples of specimens that we already had in the freezer. To give you a hint of what we're seeing here, this is now the 5x FAD amyloid model, where here we're actually using the Alamar panel to test whether we can use it for brain biochemistry. So essentially be able to multiplex changes in brain tissue lysates after traumatic brain injury. Here, what we did is we extracted the brains sequentially with guanidine after a RIPA fraction, and what we're showing here is that, again, with a fairly high false discovery rate of 50%, we are seeing some changes that are reflected within the Alamar panel on a brain tissue lysate. So this is the very first hint that we have produced within the lab that not only could the Alamar panels be useful for plasma, but then also assess, do we see parallel changes in the brain? So we're now designing studies where we're doing matched brain tissue and plasma specimens. And we think this is going to be a wonderful way in which we can then validate other methodologies, such as transcriptomic methods, to really accelerate the understanding the pathophysiological consequences of traumatic brain injury in mouse models. I'm just showing here an example of a recent spatial transcriptomic study that is just coming out in Cell Reports, where we had applied Visium spatial transcriptomics to chimera brains in mice, and we can see very clearly glial fibrillary acidic protein changes that are upregulated. These appear to be very much in the white matter, as you can see from the transcriptomic profile, as well as the immunochemistry validation of this. So the ability to incorporate Alamar profiling of brain tissue lysates as well as plasma, we have now have proof of concept that these methodologies might work, I think really set the stage for just amazing studies in the context of traumatic and other forms of brain injury. So to summarize, the field of neurology is moving at warp speed toward incorporation of blood-based biomarkers to be able to help clinicians improve diagnosis, prognostication, management, and monitoring of those with neurological conditions across the lifespan. We are really excited about the potential of the Alamar technology to accelerate discoveries along these efforts, including a multitude of clinical applications, as well as really profound insights that could be expected from animal models, where we can do the correlations between what's happening in blood and what's happening at the brain tissue level. We are very fortunate to be establishing the core facility for neurology biomarker innovation at UBC Hospital that will open officially in 2026, where we will be continuing to support a wide variety of clinical partners to measure blood-based biomarkers in their specimens and to really help guide how we might be able to implement these into clinical practice. In addition, we are very aware that the knowledge of future neurologists on these tests are coming is limited. And so we're designing a program, a workshop, hopefully that will be micro-credentialed for neurology residents and fellow to come and gain some expertise on the tests that they will likely order and how to interpret them through the core facility. And as well, internships and capacity building programs to enable medical laboratory technologists to learn the new instruments and the assays that are likely to be used, because none of this is being taught within the medical lab technician certification programs here in Canada. So with that, I will wrap up and acknowledge a number of people within my lab that have worked on aspects of blood-based biomarkers, both in clinical and preclinical settings. And we have about 120 some odd clinical collaborators, and these are just some of the major names that we've been so grateful and honored to work with. And of course, all of our funding sources that help make this research possible. So again, thank you so much for your attention today. I look forward to answering any questions, and please do not hesitate to reach out if you would like any follow-on discussions with anything that I've presented today. Thank you so much.
Oh, trying this again, just checking if you can hear my audio. I was just notified that you were unable to hear it. Okay, fantastic. So we will open up for questions now. Thank you to our presenters for their presentations. If you'd like to ask a question, go ahead and type that question into the Ask a Question box and click Submit. We will get through as many questions as time allows. So let's get started with our first question, and that question is, what sample types can I use with the Nuestra platform? And anyone is welcome to take that one. And I believe you are muted as well, Alex. There we go. Oh, nope, I'm not hearing you. It does look like you are unmuted. Do you potentially have the system muted?
Is that any better? Can you hear me now?
There we go. Yes, thank you so much.
Great, sorry about that. Yeah, so the sample types, we've actually validated quite a few different sample types with the Nuestra assay now. Obviously, I spoke about plasma and CSF, but also we've done quite a lot of work in serum and dry blood spots, and then also tissue lysates, tissue homogenates, cell culture media. So it's really possible. Urine as well. There's been a publication recently on urine samples. So due to the high sensitivity of the assay and the robustness of the performance, it seems to be very compatible with different sample types.
All right, fantastic. Thank you. Another question here. Can I build my own custom Nuestra assays?
Yep, right. We just launched at the beginning of this year, actually, a custom assay development kit or homebrew kit. So Now, if you have your own antibody pairs, then you can actually get a completely sort of easy to use kit that will allow you to conjugate the DNA molecules to the antibodies and then use the new ELISA chemistry on the Argo instrument. So we've actually had many new biomarkers developed already in the last few months using this kit. So it's possible to develop your own assays now using new ELISA.
Wonderful. Thank you so much for that. Our next question, what is the minimum sample volume required to run the new ELISA CNS panel?
Yeah, I can take that. On the Argo instrument, the instrument requires 25 microliters of sample volume. Having said that, for things like mouse samples, we have validated the assay down to 5 microliters of actual sample, so you can dilute the sample down and still get very reproducible results. So essentially, the range is about 5 microliters up to 25 microliters of sample input for plasma, serum, and CSF samples.
All right, great. Thank you for that. And thank you all for getting these questions through. We're getting quite a few. We appreciate that. Our next question, why is brain-derived, apologies if I'm saying this wrong, PTAU 217 important?
I can mention that. Maybe David has some thoughts on it as well. But what traditionally people have used assays for p-tau 217, which is really the most important marker for amyloid, for Alzheimer's disease and for actually correlating with amyloid buildup in the brain. But traditionally, people have used assays that have used antibodies that bind to the N-terminal region of p-tau 217, and that actually captures all of p-tau 217 within the blood. So that could come from muscles or from cells within the blood rather than coming from the brain. So by having a brain-derived p-tau 217 assay, we can actually measure specifically that tau protein that is coming from the brain, which is going to be very important in Alzheimer's disease to improve diagnostic accuracy. It's also going to be very important to differentiate brain-derived tau buildup versus maybe tau that's been coming from the periphery, like other areas of the body. So what we've seen is that in ALS, for example, it's a different form of tau that's actually building up in the blood, and it's not coming from the brain. And so correlated the brain-derived tau in the blood with the CSF, and you can see very good correlation, showing that that is indeed specific to the brain. So I think this is going to be very important, and the ability to look at both brain-derived in the same assay is really unique to new ELISA, and will allow us to really solve these questions, these scientific questions that have previously been very difficult to understand.
All right, thank you for that. And just to let everyone know, Dr. Wellington unfortunately did have to leave after presentation, so she's not here for Q&A, but we are going to read through the questions that were proposed directly to her in hopes that Alex or David can answer those. So this next question was specifically for Dr. Wellington. Dr. Wellington mentioned using other technologies before using Alamar and ELISA. Are those panels still very relevant, or will new ELISA be displacing most of them moving forward in the TBI field?
In the absence of Dr. Wellington, I will answer that question, from an Alamar perspective. But I believe actually a lot of these technologies are complementary. So obviously, Alamar brings the ability to look very holistically at brain health with our panels, and you can also develop singleplex assays. But I think there's so much data generated on Quanterix platforms and other platforms that it will actually be very complementary to a lot of the previous work that's been done. So I think all of these platforms have their own merits. So I think what new ELISA brings is that really high plex, high sensitivity format that enables you to look much more broadly at underlying pathology. And so I think it's unique from that perspective. And we have seen very good correlation with technologies like Quanterix and mesoscale discovery, for example, showing that we can essentially achieve similar quality data as with these other platforms. So I think each platform has its own merits, and new ELISA certainly brings something new to the market with the ability to measure all of these blood-based biomarkers in a single test, which is something that's been lacking from other techniques.
Right, fantastic. And David is logged out and come back in, so hopefully we can get him connected. I think that's him coming in now, so thank you for that. Fantastic, there we go. So Alex, we'll take some of these off of your plate. Our next question was for Dr. Wellington as well. So again, we'll pass this on to the two of you as best you can. Question for Dr. Wellington: what happens to the detectability when you use less fluid? Example, 10 microliters.
Okay, I can answer that from our perspective in terms of the validation that we've done. So in the mouse panel, for example, we optimize it for 10 microliters of input plasma or CSF, but we've also done quite extensive validation down to 5 or even 2.5 microliters. And obviously, some of the detectability of the very low number proteins will drop down a little bit, but overall, we still see greater than 90% detectability, even with as little as 2.5 microliters of input. Which obviously with mice, it's very difficult to get CSF samples, so we wanted to validate it down. But there is always a trade-off. But generally speaking, we still get very good detectability, even down below 10 microliters of sample.
All right, wonderful, thank you. Our next question: what is the main scientific driver that scientists go for Alamar instead of, I believe it's Olink?
David, if you can hear us, you can answer that, having used both these techniques. But I think the sensitivity of Alamar really gives a lot of advantages in terms of measuring a lot of the really low abundant biomarkers. Maybe, I don't know if David wants to comment.
I'm not sure, David. Yes, I'm just letting him know. He's not hearing us, but we can hear him.
Yeah, I'm not sure whether you can hear me or not. I can't hear anything at the moment. Perhaps you can put in the presenter chat if you can hear me. Okay, yeah, sorry about the confusion. I'm not hearing anything my side. But one question was question 12. So what's the main scientific driver that scientists should go for Alamar instead of Olink? So we have used Olink in the past. Olink will give us the kind of cytokine and chemokine analysis, but what Alamar will give us is a much wider range of tests, I think, and in particular, it gives us a range of neurodegenerative kind of proteomic assays. For example, the phospho-tau assays, neurofilament light, GFAP, UCH, well, a whole range of things that are not on the Olink platform. So I think part of the attraction is having a single platform that will allow us to look at both the inflammatory work and also a range of our other neurodegenerative markers.
Fantastic. Thank you so much, David, for that one. Our next question: other than human and mouse panels, any other species that this can be used for?
Yes, in fact, we've done quite a bit of work now with non-human primate samples using the CNS and the inflammation panel. And so there's quite good cross-reactivity there, and this can be used for those type of models as well. And rat also, with the mouse panel, you can utilize any rat models you may have, because the cross-reactivity there is also quite high. And so we've tested it across those different types of samples with really good quality results.
All right. Well, thank you so much for getting through all of those questions today. Unfortunately, we have run out of time, but I would like to again thank our presenters for their time today, for their important research. We'd also like to thank Labroots and our sponsor, Alamar Biosciences, for underwriting today's educational webcast. Before we go, I'd like to thank our audience for joining us today and for your very interesting questions. Any questions that we did not have time for, or those that will be submitted during the on-demand period, we will be addressing those by the speakers via contact information that you've provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues, anyone who may have missed today's live event. Until then, take care, everyone. Have a great day. Bye-bye.
Thank you. Bye-bye.